welcome to the session with Sung-Jae. I just wanted to get introducing this session, Knowledge Combined Common Data Model, a graph-based integration of Reactome and OMOP CDM. Sung-Jae, if you want to go ahead and go to your next slide, we can get started here.
I'm going to start here and okay. Hello everyone. Good afternoon from all over the world. I'm honored to be speaking here at Nodes 25. I'm Sung-Jae from NCOR, and my presentation, the title of my presentation is Knowledge Combined Data Model, a graph-based integration of Reactome and OMOP CDM. And I am a senior manager at NCOR. My company is based in Seoul, South Korea. My company runs business based on database technology. We deliver a database consulting service and some software packages for data management and data governance. And NCOR is said to be an important partner of Neo4j in Asia Pacific market area. And I was previously a research collaboration professor at Seoul National University Bundang Hospital, and which is one of the biggest hospital in South Korea. And I am also serving as an editor of ISO 29075, which is about the mathematical and statistical functions that can be used in database language standard. And this is what I'm going to talk about today. The KCDM is a knowledge combined common data model, the graph we have built. And I'm going to explain how we built the graph. And also I will explain what we can do with the graph we have built, KCDM, and also how KCDM can be further extended to be able to do further things. A human body is known to be composed of 36 trillion cells. What keeps us alive are the interactions between the cells and the interactions among the molecules inside the cells. And the malfunctioning of molecular machineries inside the cells means a disease. Clinical record and biological knowledge have many things in common, but the different vocabularies they use block them from talking to each other. And they use different vocabulary for the same thing. And the RX norm is the terminology usually used in a clinical site, and Shabby ID is the vocabulary usually used in a biological science society. And the clinical data and biological knowledge resides in separate silos. So for the interpretation of clinical outcomes, the doctor's knowledge is all we can rely on. And when it comes to the biomedical research, hypothesis generations are limited by the knowledge the researchers have in their head. We have built an integrated graph, KCDM, by combining Reactome and OMOP CDM so that the two data can interoperate. And Reactome is about the biological pathway in knowledge graph. And the OMOP CDM is the database, the standardized database for clinical observations. Reactome is a biological pathway knowledge graph, and it is open source, open access, manually curated, and peer-reviewed pathway database. And very fortunately, Reactome is provided in graph, which is instantly loadable and usable on Neo4j. OMOP CDM is a standardized data model which is designed to enable collaborative medical research across healthcare institutions by facilitating the sharing and integration of medical data. CDM deals with the whole bunch of things inside the hospital. In our work, we focus on the person and drug exposure and the condition occurrence. Condition occurrence records the diagnosis, the disease the patient have, and the concept, which is vocabulary. We have developed a unified graph structure, KCDM, and by integrating two complementary knowledge sources, the Reactome pathway database and the observational medical outcomes partnership common data model, which is OMOP CDM, containing Mimic4 clinical data. And we bridged these two data sets by mapping the ontologies. Firstly, we have downloaded the Mimic4, which is a free, open accessible electronic health record, which can be used for non-commercial medical research. And we uploaded the Mimic data onto the relational database, where we converted the data set into CDM format, and we further converted the CDM data set into a graph format, and we uploaded the CDM in graph onto the Neo4j. And the Reactome, for the biological knowledge graph, we have downloaded Reactome, and we have uploaded the Reactome onto the Neo4j. As I said, Reactome is some data resource we can use directly uploaded onto Neo4j. And on the graph, we have created the edges that can map between the nodes of CDM and the nodes of Reactome. We have found the for 1,000 chemical drugs in Reactome, we were able to find the 184 mappings in a CDM graph, and out of 745 disease code in Reactome, which is in DOID code, we were able to find 574 mappings in CDM. When we are doing this, we took advantage of biontology, which provides the mapping between the ontologies. This is the resulting graph, resulting integrated graph on Neo4j. And this is the size of the graph we have constructed. In overall, we have 33 million nodes and 64 million relations, and Reactome accounts for 2 million nodes and 10 million relations. And the rest of them is from CDM, and a few hundred edges we have added to map the data between the CDM and to the nodes in Reactome. This is the resulting graph details. We have 17 million drug exposures, which is the prescriptions for the patients, and 9 million concepts, and 4.6 million condition occurrences, which is the diagnosis event. And we have 337,000 patients and several hundred Reactome codes and chemical drugs and disease code from Reactome. This is the resulting graph schema. This side is from CDM, clinical observations. A person visits hospital, and the encounter event is recorded in visit occurrence, and the patient is diagnosed with some disease. In CDM, they use SNOMED city for disease code, and the patient is prescribed with some drugs, which is in RX norm code, and the patient undergoes some medical test, and the test result value is recorded in measurement, and the variables for the value, we use LOIC code, which is a kind of a standard ontology. And this side is from Reactome, and the Chevy ID is a drug code that is used in Reactome side, and which is mappable to RX norm, and DOID is disease code the Reactome uses, which is mappable to the SNOMED city terms, and this is the chemical drug, which is more specialized into some branded label, and it is further generalized into physical entity, and it participates in the reaction, and out of the reaction, the product is also a kind of a physical entity. With this integrated graph, we can run the queries which requires traversing through the graph. From a node from CDM graph, we can traverse through the graph to reach a node in the Reactome graph, and we can run the queries like what does the drug I medicate do in my cells in molecular level, and we can answer this kind of questions out of traversing the two graphs, and this is the information we can get from the graph, which I draw in more intuitive diagram. The drug I medicate is some signal blocker which binds to some signal receptor on some vessel cells, which controls the elevating the blood pressure. This is the cipher query show me what the drug I medicate to do in molecular level. This kind of query can be processed with this cipher query. And for simplicity, let's suppose we have Reactome code which is used in CDM side, and let this node be the anchoring node. And from this anchoring node, we can travel through the mapping edge to reach a node in Reactome side, biological knowledge graph, and we can further we can reach the reaction the drug take part in, and from this reaction we can further extend some physical entities which is input to the reaction and the product out of the reaction, and we can have the resulting graph that looks like this. And this is the anchoring node. This is the drug I was prescribed with, and which has the Reactome code in this value. And with this anchoring node, we can reach somewhat generalized drug code, which can be mapped to Chevy ID inside from the Reactome side. And from Reactome side, we can specialize this generic code into some branded drug code, which again generalize into some physical entity. This is the angiotensin receptor blocker, and this physical entity participates in this reaction, and this reaction means this blocker is bind to some signal receptor protein, and which produces a protein chemical drug complex. And in more intuitive diagram, this is the drug I have medicated, which is angiotensin 2 receptor blocker, which binds to some signal receptor, and which blocks the signal that elevates the blood pressure. This is the information we can get out of the graph. The query we can the subgraph we can have. And this is the second example. The previous example was traversing from CDM side to Reactome side, and this example is traversing from Reactome side to CDM side. This is retrieve all the patients who were treated with the drugs of angiotensin 2 receptor blockers, and this kind of query is very useful in conducting the medical researches. This is the cipher that can process this query, and this is the anchoring node, the physical entity, which has the display name of angiotensin receptor blocker, and by traversing through the graph and through the mapping edges, we can reach a node of a CDM site, and who was prescribed with this drug, and who we can list of the persons who was medicated with this an ARB as a physical entity. And this is the resulting graph. The angiotensin receptor blockers is the chemical compound I am interested in, and which can be more specialized into some branded drug, and it has some reference node for Cheviide, which is mappable from the drug code of CDM, and which can be more specialized and into a branded drug, and it is prescribed to the patients. And in this way, we can list up the patients who have medicated angiotensin receptor blocker chemical compound in our hospital. And by observing the measurement, by observing the outcomes of these patients, we can conduct some further medical research. And this is a new subgraph we can get because we have newly constructed this KCDM. And we expect this graph can be further expanded, and KCDM can be further expanded to include personal genome information and predicted protein characteristics of individuals, which can explain the drug efficacy between the individuals. And nowadays, as you know, we have the AI capabilities such as AlphaFold. We can predict the protein structure based on the personal genome sequence data. And also we have the AI-based prediction mechanism that can predict the affinity between the protein structure and some chemical compound. And by expanding this graph into genomic sequence data and further extending to this AI-based prediction data, we can explain the drug efficacy difference between the individuals. And in summary, we have integrated the two graphs. One is phenotypic level clinical observations, and the other one is the molecular level biological pathways. We were able to bridge the two graphs by ontology mapping when we took advantage of the open, free accessible mappings crawled from biontology. And with this integrated graph, it became feasible to run the queries that requires traversing through the two graphs, which is clinical observation, and the other one is the biological knowledge graph. And we expect that this graph can be further expanded to include the personal genome and predicted protein characteristics, and this will enable some molecular level interpretations of individual clinical outcomes, and this can lead us closer to precision medicine. This is my presentation, and thank you for your attention. And if I have minutes, I can have some questions. Maybe, yeah, we have 4 minutes. And if you have any questions, let me know. Okay. If you don't have right now, you can send me some email, and we are planning to make this graph, the combined graph, somewhat available for research use. You can have, I think we can have chance to use this graph. Thank you again for your attention.